Okay, so this is part one of the urinary system. So the urinary systems are the primary system that's going to be involved in excretion of fluids from the body. It is composed of a pair of kidneys in the urinary tract, which would include the bladder, ureters, urethra, and then some accessory structures. The kidneys will filter the blood to remove metabolic waste products. It will also modify the resulting fluid for the following purposes: to maintain fluid and electrolyte homeostasis, which will also tie into blood pressure regulation, as well as maintaining an acid-base balance. Now, the urinary tract is composed of a pair of ureters, a urinary bladder, and a single urethra. Urine will exit each kidney through the ureters on the posterior body wall. Each ureter will empty into the urinary bladder on the floor of the pelvic cavity, and the bladder is where urine will be stored. Urine exits from the urinary bladder through the urethra, and this will exit the body. The kidneys are the site where the urinary system regulates the homeostatic processes, and the kidneys will perform the following functions: filter blood to remove metabolic waste and eliminate when urine exits the body, regulate fluid and electrolyte balance by regulating the osmolarity. Osmolarity is going to be the blood solute concentration by either conserving or eliminating water and electrolytes. In addition, the third main function is to regulate acid-base balance and blood pH by conserving or eliminating hydrogen ions and bicarbonate ions from the blood. The kidneys will also directly influence blood pressure. If you regulate the blood volume, that will, if you guys remember, will be directly proportional to blood pressure. So if you increase the volume, you increase the pressure. If you decrease the volume, you decrease the pressure. And the way it does this is by secreting enzymes that influence blood volume as well as peripheral vascular resistance. And we kind of talked about some of this stuff with the cardiovascular system. The kidneys will also regulate red blood cell production, which is the process called erythropoiesis, by releasing a protein called erythropoietin. And it's also vital for many other metabolic functions, including detoxifying substances in the blood, activating vitamin D, as well as making new glucose through a process called gluconeogenesis from non-carbohydrate sources. That was one of those processes that was discussed in the metabolism chapter. The kidneys, structure-wise, will look like beans in both shape and color, and that's sort of where the name kidney bean comes from. Both kidneys are outside and posterior to the peritoneal membrane, so these organs are referred to as retroperitoneal. And the right kidney is slightly inferior position due to the liver, which is on that right side of the body there. The left kidney will be positioned between T12 and L3. If you're using the vertebral column as a reference. The 11th and 12th ribs provide some protection for both kidneys. And the adrenal gland, which is the gland that we talked about in the endocrine system, is located in the superior region of each kidney. So here we can see, looking at the anterior and posterior views, you can see the kidneys nestled in there. They are actually a lot higher than a lot of people think. And then you can see the ureters. These are the vessels that go down to the bladder. Okay. So let's look at the anatomy of the kidneys. Each kidney is held in place and protected by three external layers of connective tissue. From superficial to deep, they are the renal fascia, adipose capsule, and renal capsule. So the renal fascia, this is dense, irregular connective tissue. Hopefully you've seen a theme here. Most internal organs will have this dense covering, which is usually a connective tissue, and it anchors each kidney to the peritoneum and musculature of the posterior abdominal wall. The adipose capsule, this is the thickest layer that wedges each kidney in place and shields them from the physical shock. And the renal capsule, this is a thin layer of denser regular connective tissue. This covers the exterior of each kidney and it protects it from infection as well as any physical trauma. This is looking at a cross section, and here we can see the two kidneys, and you can really appreciate the fat capsule that sort of surrounds that. You can see the renal fascia and then the internal renal capsule. So each kidney is about the size of a bar of soap once connective tissue is removed, and it weighs about 150 grams. The kidneys will each have a hilum. Again, this is a structure that we've seen with the lungs and a few other structures. The hilum is the opening on the medial surface, so the midline of the kidney where the renal artery, vein, nerves, and ureters will enter and exit the structure. The hilum opens into the renal sinus. This is a cavity that's lined by the renal capsule, filled with urine draining structures and adipose tissue. There are three distinct regions that can be seen microscopically on a frontal section of the entire kidney. You're going to have the outermost renal cortex, a middle renal medulla, and the innermost renal pelvis. The renal cortex and renal medulla make up urine-forming portion of the kidney. The renal pelvis and its associated structures will drain urine from the cortex and the medulla. The renal cortex is a reddish-brown color, and this is due to a very rich blood supply. 90 to 95% of the kidney's blood vessels are located within the renal cortex. Renal columns, these are extensions of the renal cortex, will pass through the renal medulla towards the renal pelvis and house branches of the renal artery traveling to the outer cortex. Over 1 million nephrons, which are the filtering apparatus, this is the basic structural and functional unit of the kidney, are found within the cortex and the medulla. And there are two main components. What we have is a globe-shaped renal corpuscle in the renal cortex and a long renal tubule, mostly in the cortex, and a few nephrons that will have their tubules dipping into the medulla. You also have a cone-shaped renal pyramids. These are within the renal medulla, and they're separated by renal columns on either side. Each renal pyramid tapers into a slender papilla. Each papilla borders on a cup-shaped tube called the minor calyx. This is the first urine draining structure. You will then have 3 to 4 minor calyxes drain into a larger urine draining structure referred to as the major calyx. 2 to 3 major calyces will drain into large urine collecting chamber referred to as the renal pelvis, and then this will actually lead to the ureter. So you can see as you're moving from the nephrons, which are the basic structural and functional units of the kidney, how urine is going to drain away from those and then eventually lead to the ureters, where they will then travel to the bladder. So here you can appreciate the internal anatomy of the kidney, looking at the various structures there. Here's the renal pelvis, the innermost layer. Here's a major calyx right here, where you can see 3 to 4 minor calyxes draining. Here we have the renal capsule, which is the outer covering. Here's a renal column, renal pyramid. All right. You have the cortex on the outside, and this would be the medulla here. And here you can see what we have is general nephron, how it sits in the renal cortex here. This is a specialized nephron. This is active especially when you're dehydrated, and this is called a Junction medullary nephron. It's the one that really dips down into the medulla region here. So the right and left renal arteries, which are going to branch right off of the abdominal aorta, are going to supply the kidneys. The kidneys will receive about 1/4 of total cardiac output, which is about 1,200 mils of blood per minute. The renal arteries will branch into ever smaller vessels through the renal sinuses on the way to the renal columns and the renal cortex. You can use the next figure on the next slide, to trace the blood flow from the largest to the smallest arteries. So you're going from the renal artery to the segmental artery to an interlobular artery, a cruate artery, and then finally the cortical radiate artery. So the kidney contains unusual capillary bed system. Arterioles both feed and drain the capillaries. Normally, that's a function that we see the draining aspect of a venule. Each interlobular artery leads to an afferent arteriole, which feeds into the ball-shaped capillary bed. This is the first capillary bed, referred to as glomerulus. The glomerulus and its capillaries are part of the renal corpuscle of the nephron, and they will actually drain into an efferent arteriole. The efferent arteriole will then feed into a second capillary bed of the nephron, referred to as the peritubular capillaries. The peritubular capillaries will form a network or plexus surrounding the renal tubules of each nephron. So you have the afferent arteriole will feed into the glomerulus, which drains into the efferent arteriole, which will feed into the peritubular capillaries. The venous blood exits the kidney parallel to the arterial pathway, beginning with the peritubular venules that drain the peritubular capillary beds. The peritubular venules will unite to form a progressively larger venous vessel. And you now have the interlobular veins, which will drain into the cruate veins, into the interlobar vein, and then back into the renal vein. The renal vein will then exit the kidney from the hilum, and then that will drain into your inferior vena cava, which will go back to the right atrium of the heart. So here you can see the blood supply from the renal artery to the segmental artery to the interlobar artery. And then you have the cruate artery, which is going to feed into one of these little networks here, then into the interlobular artery, which will feed into an afferent arteriole. The glomerulus is your first capillary bed. Then you go out through the efferent arteriole, and then the efferent arteriole will feed into the peritubular capillaries, where you'll go back in through the cruate vein, interlobular vein, and then back into the renal vein, which will feed back into the vena cava. So the nephron, there are two main divisions. You have the renal corpuscle and the renal tubule, and they're collecting subdivisions. The function of the renal corpuscle is responsible for filtering blood, and it's composed of two parts. The glomerulus, which is a group of looping fenestrated capillaries. These fenestrations are quite prominent, and that allows the capillary bed to be extremely leaky or permeable. Then you also have the glomerular capsule, which in my day was called the Bowman's capsule. And this is a double layer outer sheath of the epithelial tissue. And it consists of outer parietal and inner visceral layers. So this is this area right here. This would be the renal corpuscle. So the glomerular capsule, this is a globe-like parietal layer, which is an extension of the renal tubule, made up of simple squamous epithelium. The visceral layer is made up of modified epithelial cells, referred to as podocytes, whose special extensions surround the glomerular capillaries to form filtration slits. Essentially what's going to happen is you have the capillaries within the glomerulus where filtrate, which is fluid from the blood, will exit, and it will pass through those little slits, filtration slits, and be captured by this glomerular capsule. The capsular space is the hollow region between the parietal and visceral layers, and it's continuous with the entry into the renal tubules lumen. So what's going to happen is that fluid collects within the capsule. It will then start to feed into the tubule system, where you will have the opportunity to reclaim things that were excreted, as well as excrete things that the body really wants to get rid of. The podocytes in the fenestrated glomerular capillaries will form complex membrane that filters the blood flowing through the glomerulus. The filtered fluid, which is referred to as filtrate, exits the glomerular capillaries into the capsular space, and then it enters into the lumen of the renal tubule. So this is our glomerulus right here. Okay, so what you see is here's an afferent arteriole. This is feeding in. This is a capillary bed here. Okay, and there's going to be pressure differences that are going to allow fluid to leave the capillary and enter into this space here that's formed by the glomerular capsule. Okay. The visceral layer is this tan area that you see on the actual capillaries. And you can see these little slits that are formed by that visceral layer. Okay. This is the parietal layer here, which is going to form the outer walls of the capsule. And then you have the space here that the filtrate will accumulate in, which is referred to as the glomerular space. What will eventually happen is that filtrate will now, that has been separated from the blood, will now go through the filter of the tubule network. And here's an EM electron magnification looking at the capillary, and you can see the visceral layer with this filtration slits. So the newly formed filtrate enters into the renal tubules, where it can be further modified in 3 structurally and functionally distinct regions. The first is the proximal tubule, then you have the Nephron Loop, and then the distal tubule. So the proximal tubule, this is the first and longest segment of the renal tubule. Both straight and coiled or convoluted sections exist. It is made up of simple cuboidal epithelial cells with prominent microvilli. Now, whenever you see a cell that has lots of microvilli, and we talked about this with the digestive system, is that this will allow those cells to function in absorption. And that's what we're going to see here, is you're going to have lots of absorption of materials back into the blood, essentially by those peritubular capillaries that are sort of surrounding at that point. The Nephron Loop, or the Loop of Henle, this is the next segment that filtrate enters into. It is only part of the renal tubule to dip into the renal medulla, and it consists of a descending and ascending limb. The descending limb is known as the thin, is known as a thin descending limb because it's only made up of simple squamous epithelium, as you can see in this figure right here. As you can see in this figure right here. Okay. Once a descending loop turns 180 degrees, it is now known as the ascending limb. And this is sometimes called the thick ascending limb because it is now made up of simple cuboidal cells again. Some nephrons will have a thin ascending limb at the bend because the region is made up of thin, simple squamous epithelium instead of the thicker, simple cuboidal cells that are typically found a little more upstream. The distal tubule, this has both straight and convoluted sections. This is the last segment of the renal tubule that filtrate passes through, and it's made up of simple cuboidal epithelium without any brush border. So you want to know what looking at the four distinct regions here, the proximal convoluted tubule, the thin descending limb of the loop of Henle, the thicker ascending loop of the loop of Henle, and the distal convoluted tubule. Make sure you know what the histology looks like at each of those areas. We also have another structure which we refer to as the Juxtaglomia apparatus. This is composed of both the macula densa and the JG cells. This is found at a transition point between the ascending limb of the nephron and the distal tubule. The macula densa, this is a group of cells in contact with the modified smooth muscle cells, which is referred to as JG cells, in the walls of both the afferent and the efferent arterioles. What the JG cells are responsible for is actually monitoring the blood pressure that is going through those vessels, as it is very important that you need to maintain a certain blood pressure, not too high, not too low, so that you can get appropriate filtration production. And right here, you can see this would be the smooth muscle in the afferent and efferent arterioles, and these green cells here would be the macula densa. The final segment of the nephron is going to be the collecting system. And this is a series of functionally and structurally distinct tubules that consist of a cortical duct and a medullary collecting system. Both the medullary collecting duct and the papillary duct. They will further modify filtrate before it actually exits the kidney. And when we say modifications, we mean that you can affect the alterations affect certain concentrations of things that are present in that particular filtrate. Now, typically, any modifications that occur at this point in the filtration process is going to have to be hormonally regulated. So the things will move based on hormone concentrations that are present. So once filtrate enters into the papillary ducts, leaving the collecting duct here, what will then happen is now it's no longer referred to as filtrate, but now it's referred to as urine. Okay. Urine will then exit the papillary ducts at the papilla of the renal pyramid and go into the minor calyx. So the kidneys will contain actually two structurally and functionally different nephrons. Cortical nephrons, this is the most common nephron. It's also the most abundant nephron in the kidney, as well as juxtamedullary nephrons. The juxtamedullary nephrons will actually dip down into the medulla region of the kidney. So your cortical nephrons, they make up about 80% of the nephrons in the kidneys, and they are primarily located in that cortical region. The renal corpuscles, these are located in the outer renal cortex, they have a short nephron loops that barely enter into the renal medulla, if at all. You also have the peritubular capillaries, which are going to surround this tubule network. The reason for those peritubular capillaries, as blood is flowing back, you can put things back into the blood through these capillaries and continue to remove things from the blood that the body wants to remove through this whole process. The ducts and medullary nephrons, these are much less common than the cortical nephrons. And the cortical portion of the nephron is actually surrounded by peritubular capillary branches from the neighboring cortical nephrons. The nephron loop is surrounded by a ladder-like network of capillaries that has a different name referred to as the vasa recta. And these arise from the efferent arteriole. This particular capillary is unique because of the structural arrangement that allows the juxtamedullary nephrons to control the volume and the concentration of urine. This particular type of nephron is really used when an individual is dehydrated. And here you can see the difference between the cortical nephron here versus the juxtamedullary nephron here, where you can see it's much more deeper into that medullary region. And this will be the end of recording number 1.